So the last two slides here, and I think this is kind of what I said to you guys on Thursday, that I have the actual, in actuality, I have the same bullet point at the end of both of these slides. And the question is essentially, how do we make sense of parts of the brain that we typically associate with sort of non social behavior, making contributions to judgments that are social in nature, or broadly speaking, social behavior. And I was being a little bit facetious in truth, because um, the way we've been talking about neurology and localization of function and specialization throughout the course has really tried to get you to sort of confront some of the commonly held beliefs that we have, for example, a faith center in our brain, or we have a social center in our brain, which intuitively makes sense because we know there is a great deal of specialization. So it does kind of fit with how we think about other parts of neural organization, like the motor cortex, an area that's specialized for movement. Why wouldn't there also be an area that's specialized for socializing and making social judgments about people in social situations, social contexts? But as I said, as I've been saying throughout the course, and as I'm sort of building up into for these last two slides, I think that it's sort of a mistake, and you can become a victim of confirmation bias if you assume there must be a social region of the brain, and that's the seat of all social cognition. So really, in many ways, this idea that this part of the brain that is normally involved with perceiving physical temperature, physical warmth, this part of the brain contributing to social judgments about interpersonal warmth seems a bit peculiar on the surface of it, but in many ways, it's not any different than the TPJ maybe being associated both with shifting your attentional set, as well as the theory of mind kind of representations that we talked about. Usually things that are products of evolution in biology are particularly efficient. And in the brain, we know that most regions, maybe we could argue all, but that's maybe not, we don't really have to come up with a definitive bottom line there. But I think we could entertain the argument that no parts of the brain are specialized for only one thing, that really they all are contributing in more generalizable ways. So this is a sort of straightforward study. If you hadn't read it before, if you hadn't read the slide before today, you probably read it now since I've been talking. But it sort of looks like there's this almost spillover effect, or this signal that is usually associated with one thing, that almost, we can think of it a little bit like it almost bleeds through. And so I've used this terminology before, but it's almost like it contaminates another signal. Like you have a judgment you're forming about how warm they are socially, which is presumably based on the way they're interacting with you, the things they say to you, maybe facial expressions. But you also have this signal coming from another part of your brain that, again, is almost, it's as if it's bleeding through or contaminating the other signal that is coming together based on many parts of the brain, no doubt sending their own signals to aggregate a decision. It's also, the last thing I'll mention is one kind of cool thing about this is it reflects what we could consider an everyday sort of cross-modal like association, or cross-modal like mapping. So synesthesia is something that I know you guys are familiar with. We've talked about it briefly already in our course. It'll come up in consciousness again briefly. But it's this idea that, for example, maybe every time you see the number 5, no matter what color ink it's written in, you perceive it as being blue. So these kind of unusual cross-modal associations, they're not always cross-modal. What I just said, color grapheme synesthesia, that's all within the visual modality. But often there's a crossover. So you, for example, will experience a certain taste or smell when you see certain colors, or you'll see certain colors when you hear certain pitches or notes. So this is very fascinating for a whole bunch of reasons. And we actually saw a really clever use of a synesthetic population to more specifically test a hypothesis back when we were looking at one of the memory chapters. But it's also the case that in the general population, the average person has what we could consider cross-modal associations that are sort of illogical. So people tend to, if I give you a really low, bassy, rumbly note, and I give you a high-pitched, bright, twangy, trebly note, and I ask you to match those with colors, people typically match the low, deep, bassy notes with dark, rich colors, and the higher pitched, treble heavy notes with lighter, brighter colors. And so there are examples like this where we think people are predisposed to kind of have an equivalency. If you think back to the idea of common currency, which we talked about in decision making and value learning, value judgments, there's this idea that you have to be able to equate apples and oranges. They're different things, but you have to be able to equate a value with each. And we can think of a similar process here, where we have this sort of way that we equate qualitatively different things, and we get, this specific example might not be particularly adaptive, this behavior, but we get glimpses into the kinds of ways we associate different things that may be potentially qualitatively very different, based on these kinds of associations. It also, so that I've said a lot about this already, the last thing I could say is, in a lot of cultures, it's a ritual. You have a tea with someone. Tea ceremonies are in certain parts of the world incredibly important. You go out for a coffee with your buddy. You catch up with a long lost friend at a cafe. Hot drinks are actually a part of our social behavior in many contexts. And you have to wonder if, not that this drove that, but if maybe this just helps them fit into that kind of context that much better. So again, on this slide, we have an example of an experiment that's showing you parts of the brain that we could argue are at least not exclusively involved with social cognition, that seem to nevertheless be generating a signal that's guiding social behavior, or at least social judgments. So we see the ACC come up again. We've been talking about that guy a lot recently in the context of error signals, error-related negativity. And we see that in this simple experiment where they do a virtual game of keep away, essentially, where there's one computer, or there are two computer players. These are kind of like confederates, actors that are part of the experiment, but the naive experimental participant who's not aware of the purpose of the experiment thinks that they're just other random participants, just like themselves. So these two computer players don't include the actual human player. And again, they're using computers. It's a computer-based game, but there is an actual human player, of course. They're also being scanned. So with fMRI, we're looking at ACC activity. And we get people to report a sort of subjective judgment about how sort of hurt or left out or distressed you felt by being left out. So totally subjective, right? It's a behavioral measure, but then we also have ACC activation. And we see that there's actually a really reasonable correlation between magnitude of the bold signal or the change in the bold signal related to the ACC and the extent to which their judgment reflected someone who basically had their feelings hurt. So in other words, people who self-reported that their feelings were hurt more showed greater ACC activation, and people who reported that it didn't bother them, they didn't really care, didn't really show much ACC activation, at least relative to the first group. So this you should be able to put into the context of other kind of error signal stuff we've talked about, which is cool because it among other things, shows you this kind of generalizable neural mechanism, an error signal that can be used to guide all kinds of behavior, right? Choices, forced alternative choices between two things that you might value to different extents. When you're, the last experiment we talked about, or one of the last ones last time, where you're watching another player take their turns in a game that you're also playing. And we see that error signals, the researchers hypothesized, were used to model not only the expectations for your own actions and your own rewards, but what you were expecting the other player to do and what rewards or outcomes or consequences you were expecting the other player to receive. Because of the choices they made, we can build up a lot of kind of complicated behaviors. We can model a lot of kind of complicated behaviors, like something as arguably nuanced as having your feelings hurt when you're left out, by thinking about how error signals might be a part of the system that kind of regulates that. Final kind of thing to note about this, probably a lot of people, well, let me rephrase that. Probably for the average person, being left out of a computer-based game when you're doing an experiment, when you might be suspicious that, that's part of the experiment anyway, probably doesn't hurt your feelings as much as if you're playing a real game with friends, people you actually care about. So we could imagine that this is a very reasonable way to set up the experiment, and they clearly get interesting results that we can dig into and make some sense of. But if anything, this might be kind of underestimating the effect. Maybe what looks like a, sort of mild-ish effect might actually be much larger in a more real-world context. And of course, those two last experiments were sort of neat segues into talking about emotion this week. Affective neuroscience is one kind of more formal way to term that, by affective neuroscience. So your affect is like your mood, right? If you're in a good mood or a bad mood, if you're stressed out, if you're relaxed, that's all related to your affect with an A. So affective neuroscience is the branch of neuroscience that specializes in trying to understand the underlying physiological and neural mechanisms that drive those kinds of behaviors. Now, I made the point, I think maybe last chapter when we started social behavior, about how some of these last few chapters overlap a fair bit. And in addition to having somewhat common mechanisms, or at least mechanisms that we could speculate might be contributing to multiple things, like the ACC and an error signal that shapes your social behavior, but also shapes your decisions about which rewards to choose. There's potentially a lot of overlap. And we're also getting into a situation where, in many ways, we're following the textbook structure, or the back half of the textbook structure, which kind of basically goes from, in some sense, simple to complex. And the last few chapters get pretty complicated, and in many ways are much more difficult to operationalize and study scientifically. We can't directly measure memory. We still have to make an indirect inference about memory by doing things like giving people comprehension tests. And there are reasons why you might get a question wrong on a comprehension test, even though you actually remembered it. It's not a perfect measure of memory, but in some ways, it's a reasonably direct way of getting at memory, we could argue, getting at making an inference about memory. On the other hand, when you start trying to operationalize things like trustworthiness or social behavior or emotion, I think the potential issues that come up in kind of the early design stages, we could kind of spend longer talking about how maybe, and critiquing potentially, how experiments are set up to target things like emotion or social behavior. For example, when we talked about decision making, the first chunk of it, the first third of it, was really about perceptual decisions, which gave us some insight into, for example, parts of the brain that control eye movements that seem to predict decisions that are being made about what people are perceiving or what non-human animals are perceiving. But we could argue, is that really the same as what the average person means when they talk about, I wonder what guides my decision making? They're probably talking about other things. And even though the two alternative forced choice value learning stuff was a bit more complicated, probably the average person is still talking about something other than, choosing one of two things that pop up on a screen over many repetitions. That's quite artificial and contrived, of course. So one of the kind of warnings here is, and it makes it kind of fun potentially and a little more exciting, because I think you have to do a little more work just to kind of frame these concepts, let alone actually collect the data and interpret the results, which is another hurdle that we've spent a lot of time focusing on. But one of the cool things about emotion is it does overlap and dovetail and make contact with all kinds of other areas of cognition. We started off the class, arguably, we had a sort of historical section where we gave you a bit of background and reviewed some neuroimaging techniques. But really, the sort of first topic that we tackled was object recognition, with a focus on facial recognition. And that's something that, of course, and we're going to see more face processing in this chapter, in this through this week, through our 2 lectures this week. But that is very much something that can be influenced by emotion. Think about how different you feel when you recognize like a loved one's face as compared to a stranger's face that you decide you don't recognize. Emotion affects decision making. Emotion affects cognitive control. Think about how much more difficult it is to stifle an impulse, to restrain maybe saying a mean comment that you know you shouldn't say, but you kind of feel compelled to, if you're tired or if you're stressed out. Emotion kind of colors really all of the kind of cognitive stuff we've talked about thus far. So it's really very valuable to study, in addition to kind of coloring what we would call, what we could term more kind of typical normal population cognition in various aspects. It also has a huge importance for clinical stuff. A lot of clinical problems that people have can be understood, at least in part, as being related to a dysfunction or a dysregulation of emotion. So it's a very important topic, in addition to being something that is interesting to people. It's sort of, I'm being a little bit meta here, but it's people talk about things that are evocative as being things that are emotional, that induce an emotion in you, are evocative. And just studying what makes us feel the things we do in and of itself is kind of emotional, right? The feelings that we feel that are so profound and so important to us and so central to our lives, the things that we feel good about, the things we feel bad about. It's quite something to try to reduce that to neuronal firing. So for this lecture today, we're basically going to be focused on the amygdala. And I'm sure you've been hearing since intro psych that the amygdala is part of the limbic system. It's important for emotion. And that is absolutely true. We've got a kind of seasonally appropriate graphic here with a jack-o'-lantern. And fear-based responses and behavior that has to do with fear and fearful responses to stimuli are in particular something that we very closely associate with the amygdala. And we could imagine that a lot of, you're probably aware that people have historically argued about how many fundamental dimensions of emotion there are. Some people argued that there are 20 different fundamental base emotions. Some people have argued there are 3. Lots of people have argued numbers in between. And so in some ways, we can kind of carve up emotions in lots of different ways, right? I could think of lots of synonyms for happy that we could maybe mull over. Are they unique feelings? Is pleasure and happiness and joy and elation, are those just different words for the same thing, or are these slightly different emotional states? So sidestepping that kind of problem, which is nevertheless a methodological issue for people who study affective neuroscience and the psychology of emotion in general, we can consider a lot of emotions, a lot of negatively valenced emotions that relate in some way to fear. So things like anxiety and maybe even sadness and other kinds of emotions that having a fear-based response might be more likely to trigger, even if we think of them as somewhat distinct emotions. So I don't want you to think of, even though we keep talking about the amygdala and fear, think about how generating what we could think of as a fear signal could interact with other kinds of emotions, and in particular, other kinds of negatively valenced emotions. So in emotion, we often talk about valence, positive or negative. Emotions can generally be classified as having a positive or negative valence. In fact, in some ways, that's one of the ways people kind of operationalize what an emotion is. It's part of how some people define emotion. Then you can also talk about arousal, which is sort of like intensity. You can have a mildly negative emotion that's not very intense. You can have a very intense negative emotion. And then you can fill in the other 2 sort of cells on the positive valence side, right? You can have a really strong, intense positive emotion and a weaker, kind of more mild positive emotion. And of course, there's everything in between. But kind of what I'm getting at is there are lots of ways to sort of set up the framework for defining and operationalizing emotion. And fear and the amygdala's role in a fear-based signal is going to be really central to a lot of these negatively valenced emotions, which also influence some social behaviors, as we'll see in our first experiment on the next slide. The last thing I wanted to do is just touch on a little bit related to, the medial temporal lobe. MTL is the abbreviation you saw over and over again in the memory chapters. Of course, the amygdala is part of the limbic system, like the hippocampus. And so the amygdala is going to be really important for memory, in addition to sort of on the spot, in the moment appraisals of things like fear. It's also the case that the amygdala is going to, we'll have kind of some question as to whether the amygdala is specialized for processing in one modality. And in particular, it seems to be the case that the amygdala is really important for responding to visually based stimuli. And in some instances, it seems to be the case that the amygdala is less relevant for processing signals that may or may not embody fear, or may or may not be related to fear, associated with fear in other modalities. And without being anything conclusive, of course, we can kind of think about the fact that the amygdala is actually quite close to inferotemporal cortex, which of course has things like the fusiform face area. So this is just some background as we think about this question of whether the amygdala is sort of an amodal, or at least biased in some way towards one modality, kind of fear center. So this is the first experiment that I just alluded to. So we're starting off with behavioral data. One of the things that you might not always have done when people show you data and experiments, but that I think is useful in our class in particular, perhaps, is distinguishing between what's behavioral data and what's imaging data. And then we can also distinguish between things like case studies and normal populations. Because as we've seen, a lot of these arguments take their strongest forms when you get nice converging evidence across different kinds of studies. If we only ever had imaging data, and of course, imaging studies often incorporate behavioral data, right? There are often responses that have to be made in fMRI studies, not always, but it helps us build up a more complete picture, and kind of having some sense of, well, have they kind of filled in all of the, is there evidence in all of the kind of typical ways they look for evidence? Having some sense of that, or is it the case that there's a huge amount of imaging data but very little behavioral data, or vice versa? Or is there a huge amount of data dealing with case study patients but very little with typical populations? This kind of thinking is not specifically relevant just for this chapter, but I think is a good thing to help you try to keep straight what's done in which studies, and have a sense of how, in combination, multiple studies that do different things can make a more or less compelling argument. So we're starting off with some case studies. So this 98 paper looked at 3 patients who had pretty significant amygdala damage. So bilateral, so both hemispheres. And what they're doing is they're rating trustworthiness of a bunch of face stimuli. So this, of course, makes contact with the question of trustworthiness in a recent topic that we just talked about, the last one. We can, think a little bit about operationalizing trustworthiness and how that may be interpreted differently by different people. We could go down the kind of methodological road. I don't want to do that just now. But what they did in brief, and so it's maybe, we're seeing a lot of bars here, in part because there are only 3 participants. So they're plotting out the data individually for all 3 participants. But really, this is maybe a simpler result than it looks like. Really, all they're saying is these 3 patients that had amygdala damage didn't seem to discriminate very much in terms of trustworthy. They kind of just thought everyone was basically trustworthy. And the sort of important comparison is they had normalized ratings. You have to do that in most cases. You could just collect a bunch of faces, but what if you just happen to collect all faces that the average person thinks are trustworthy or not? So you want to collect a data set and test, normalize your stimuli, show it to a bunch of people, get them to rate it, tweak it a little bit. If you have very few faces that the average person thinks are not trustworthy, rebalance it a little bit. And you end up with a stimuli set that that in the general population, people express variation in the extent to which they trust each of those faces. And so this absolutely veers into the judgmental territory. And we're going to talk a little bit this lecture about racism as well. Because really what this is saying, of course, if it's not obvious, is based on nothing more than the way someone looks, these people are making judgments about how trustworthy they regard them. Albeit, they maybe, this is nothing about whether people are actually changing their behavior on the basis of the trustworthiness. We saw some effects related to that in the last lecture. But this is just saying, all things being equal, you don't have any information to go on except for their visual appearance. How trustworthy or not trustworthy do you think these people are? And so what we see is, as I say, these people with damage don't seem to differentiate. And we could argue that in the average, the normal, the healthy population, the fact that some of these faces are singled out as not being trustworthy isn't necessarily a good thing. Maybe it relates to a prejudice people have. It's probably, it's not that we're saying that this is an appropriate response necessarily or a socially acceptable response, but we're saying this is how the average person responds. They judge based on appearance what they think about how trustworthy the person is. And without a healthy amygdala, a normal quote unquote amygdala, we don't see this same kind of effect. So for this next study, they used a similar kind of procedure. They had people make judgments, the participants made judgments about how trustworthy they regarded various faces, face stimuli. And one of the things that they did that is sort of methodologically relevant here is they had sort of a cover story. They didn't tell people initially up front, we're getting you to make judgments about how trustworthy you think people are based on the way they look. You could imagine just hearing that alone might make someone nervous. They might worry that they're judgmental, worry that they're prejudiced, and that could introduce a whole host of confounds, right? Better in many ways, we could argue, for the participants to remain quote unquote naive, to not know the purpose of the experiment, to not have anything to bias them going into the experiment in the fMRI session, the scanning session. So they told people that it was just an experiment investigating memory for faces. So the idea is they're not predisposing people to engage in a particular kind of task. They are predisposing people, of course, to try to encode the faces, right? It's not completely neutral. And if we wanted to, maybe we could raise some concerns about that, even just, as a remote possibility. But it is, I think, it's easy to argue that this is better than putting people in the scanner and from the get-go saying, I want you to judge how trustworthy you think these people are. So imagine, I get my first face and my initial thought is, I don't trust that person. Then I start worrying that I am a bad person because I'm being prejudiced or judgmental, right? You can already imagine tons of brain activity is maybe starting to be picked up by the fMRI that isn't necessarily directly related to their research question. And it could be, depending on what aspect of these judgments they're looking at, but they wanted to take sort of an unbiased recording of just what the brains kind of more or less naturally do when you're looking at faces. And then in the second phase of their experiment, that's when they took the behavioral measures. So if it was the case that you became self-conscious, for example, about why you were assigning the judgments you were, that won't contaminate the fMRI data because it's in a separate session. And in some ways, doing this might make it less likely to find an effect. You might think that they have a better chance of finding an effect if they actually did the judgments in the scanner, possibly. But if they separate the judgments from the scan session and they find that one part of the brain is reliably associated with the judgments, and the activity of that one part of the brain that was, active during the scanning session before any kind of explicit judgments were made, if that actually is a good predictor of the judgments they're making, that in some ways makes a more compelling case, because you've again kind of um, you've kept the neural activity during scanning slightly more natural and less, you haven't biased it in a way that you might otherwise have. So they have this really bizarre finding that kind of makes perfect sense if you remember that the amygdala is just one signal that's being combined with other signals, which eventually produces a decision, a judgment. But on the surface of it, it sounds kind of wild. It sounds like, pseudoscience, or like, like it's a metaphysical impossibility or something, or a physical impossibility. It sounds like it has a metaphysical element to it almost. When they look at the correlation between the amygdala activity at the individual level. So I'm an individual, I'm put in the scanner, I'm, I've made judgments at some point during my experimental session, and we're looking at my amygdala activation in that moment and how closely my amygdala activation predicts my judgment of how trustworthy I think the face is. There's less of a correlation there, as compared to the group average rating of the trustworthiness, and the group average of so the amygdala activity. So what they're saying here, and again, you sort of say, well, how does that make sense? How is that even possible? How could it be that my neural activity is not as good a predictor of my decision as the group activity may be? And the average group activity of the amygdala is of the judgments. And so one way to understand this, and open to interpretation perhaps, albeit one way to understand it that the researchers tried to argue, that I think makes sense when you stop and think about it, is again, the amygdala is having, we think, kind of a rapid and fairly automatic response, maybe based on prejudices, but you're picking up on something related to visual features, and you've got one signal. And you could imagine if you have, maybe if the amygdala gets really active, that happens when you see something that you're afraid of. And maybe that'll result in you, on average, providing a trustworthy rating that's lower, as compared to when your amygdala does not become active and you do not have that fear-based response. That makes sense. But then that's not going to explain all the variance in your judgment, because that's just the starting point. In theory, that initial kind of rapidly, fairly automatically derived signal then gets combined or integrated with other signals. So for example, if I have a memory of a friend that had a face that looked similar to the one on screen, maybe they have, and I'm being kind of silly here, right? But like, maybe you think about villains in old Bond movies, villains in old Bond movies have had eye patches. And so maybe, the average person sees an eye patch and the only association they have is with a Bond villain or a pirate. But what if you had a friend that wore an eye patch, right? You might have this initial rapid fear-based response in the amygdala because you associate this with Bond villains. But then you remember your friend that had that same characteristic. And then on average, you end up judging the face to be very trustworthy because your memory of your friend trumps or kind of overpowers the other signal. So sort of what this is, part of what this is getting at, in addition to just kind of a weird, illogical sounding on the surface finding, part of what this is getting at is that again, the amygdala is contributing some kind of initial input, contributing a signal, like, not that it is an error signal, but like we talked about the error signals at the beginning of the lecture, there's some kind of signal that is a base that's worked on, but then other signals get integrated and combined and factored in. So this is part of the takeaway from this, is that your amygdala activity by itself is not a good predictor in the moment, on a subject by subject basis, on a trial by trial basis, of the trustworthiness that you ascribe to that one stimulus. But on average, it becomes a very good predictor on average of the variability of these kinds of trustworthy judgments. Again, indicating that it's not the whole story, but it does factor in. So we're now going to talk about this thing called the implicit association test, an IAT. So I've linked to this YouTube video. So feel free to watch this if you're still a little bit fuzzy about this after we go through it. But I'm going to try to walk you through kind of my explanation of how these things work. And I think these are, I remember this was actually as an undergraduate when I did my undergraduate degree in psychology. In my intro psych class, of course, I had to participate in experiments, and I participated in one of these. And it was an IAT that was geared towards gauging racism. How much unconscious implicit bias do you have related to races, related to ethnicity? And this is in many ways a very controversial test. People argue about whether or not it measures what it's supposed to. And the bottom line is, in theory, it quantifies how racist you are, which is this isn't how anyone who is using the test scientifically would describe it. They'd be much more careful with their language. But I think this is one of the reasons why it's so contentious, because people have very strong beliefs about whether or not they have biases. And one of the kind of tricky things about biases is people aren't always aware of their biases. So part of kind of what I want you to be thinking about is, I don't want you to take this at face value as perfectly measuring racism, because it probably doesn't. And there are probably some problems with this. But, think about it critically, and, don't assume that it's got some value, and consider what you think about it. Consider whether you think that this is a defensible way, whether it makes sense that the results they get, the effects they measure, they interpret as relating to implicit bias. So I've got kind of a lighter example, depending on how you feel about Canadian-American relations. But we're going to look at how this would work if we were trying to measure implicit bias towards or against Americans or Canadians. So I'm going to stumble over my words if I try to kind of read these out in sequence. So I'm just going to explain this kind of in my own words, even though the slide's in my own words, I might jump around a little bit. But you've got this handful of essential components. You end up making judgments about valence, so good and bad, and they may throw out some synonyms. You might be judging like reward, punishment, desirable, undesirable, attractive, unattractive, good, bad. I'm just going to use good and bad to keep it simple. But they get you to make multiple kinds of judgments with 2 response keys. And that's really kind of critical, that you only have 2 response keys, because the whole point is you come to associate certain things with certain keys. And because you're making multiple kind of judgments about multiple things using only 2 response keys, the idea is you end up getting something similar to priming effects, or compatibility effects. So imagine you start off and you're shown things or words, right? You can set this up in lots of ways, and you have to identify good or bad using, for example, the left and the right shift button. So imagine if it's good, I press the left shift button when it appears on screen. If it's bad, I press the right button when it appears on screen, the right shift button. So maybe I'll see things like, like the word love and the word war and hate, and things that people would have a fairly consistent valence categorization for. So that's the first piece. Now, the next piece is the thing that you are kind of actually investigating as your target of focus, target of the experiment. The implicit attitude that you're trying to measure is the next thing that we need to talk about. So if I'm trying to measure implicit attitudes towards Americans, implicit biases, for example, towards Americans, there'd be some trials where I had to identify, again, stimuli, whether it was word, whether it was a picture. I'd have to make a response to things that flashed up on screen and essentially categorize them as Canadian or American, right? So maybe you'll see we could, maple syrup, maybe Canadian would be the right response. Maybe I'm trying to think of all the things that I think of as being quintessentially American, we kind of associate with Canada too. Like I was going to say beer, but people think of beer as being part of the Canadian culture. But if you have president, right? Keep it simple. President, you press the right button because that's something you associate with Americans. Prime Minister, you press the left button. That's something you associate with Canadians. So we've got two kinds of judgments that are both using the same two response keys. So then the thing that happens is you reverse the mapping. So if you had been pressing the left shift button to kind of tag something as being